Dr. Rosso teaches a class every Tuesday afternoon from 3 to 5, so he's not able to join us today. So as the lab manager, he asked me to introduce Dr. Saeed El-Shafei, which of course is an honor for me. Dr. El-Shafei received his bachelor degree in veterinary science in 2005 from Benha University in Egypt. Should I mention warm Egypt? Today they had a high of 64 degrees, and on Monday they're expecting a high of 74 degrees. Did I mention that it's also winter there right now? It's depressing for us, not for them. Anyways, Dr. El-Shafei was promoted to assistant lecturer in the pathology department of the College of Veterinary Medicine at the Benha University in 2006. In 2009, he obtained the master's degree, and during that time he received numerous awards. He's gotten awards from the Egyptian Society of Anatomy, the Egyptian Society of Histology. He won the Best Presenter Award at the third conference of the Benha College of Veterinary Medicine. In 2012, Dr. El-Shafei joined the Dr. Rosso lab, and he passed his candidacy exam in 2014. He has several projects that he's working on in our lab, and they include doing histology studies on dog prostate. He's developed a novel canine prostate cancer cell line. He's been looking at microRNAs in prostate cancer, as well as doing a study on evaluation of AR42 as a potential anti-cancer agent in prostate cancer. However, today he will be speaking to us about his project studying the GRPR signaling in prostate cancer. So Saeed, we look forward to hearing about your exciting research.
Thanks, Russell, for this nice introduction. I should say that your introduction was more detailed than my presentation would be, but that's okay. So my topic today is about the role of GRPR signaling in prostate cancer. And so before going and discussing my data, I'd like first to give you an introduction about GRPR. So what's standing for? It's standing for gastrin-releasing peptide receptor. It's a subtype of bombesin receptor subfamily. It's a bombesin receptor type 2 receptor. It belongs to G protein coupled receptor superfamily. And it's a glycosylated 7 transmembrane receptor, which means it spans the cell membrane 7 times, creating 3 extracellular loops and 3 intracellular loops. It has N-terminus, which is located extracellularly, sometimes named ligand binding domain, and C-terminus located intracellularly and sometimes named as G protein subunit domain. GRB is a natural ligand for GRPR and regulates many functions in the body. For instance, gastrointestinal hormonal release, smooth muscle contraction, has a mitogenic effect on epithelial cells. Also, it acts as neurotransmitter in brain and growth factor in developing brain. And some studies, they found that GRP play a role in lung development. So GRPR as a receptor is normally expressed in just few organs in the body, for instance, stomach, pancreas, adrenal cortex, and brain. But it's abnormally expressed in many tissues in the body in case of cancer, like lung cancer, prostate cancer, and the colon cancer. So you may appreciate my work if I told you that GRPR is not only highly expressed in about 90% of prostate cancer, but also expressed in 75% of pancreatic cancer, 90% of lung cancer, 50% of breast cancer, 100% in head and neck cancer, and 85% of brain tumor, especially glioblastoma. So what is the relationship between GRPR and prostate cancer? So most studies found that there is higher GRPR expression in both PIN, as it's a prostate intraepithelial neoplasia, which can be considered as early stage of prostate cancer, and also in invasive prostate carcinomas. And they found also that GRPR modulates tumor cell proliferation, appearance, and differentiation. And activation of GRPR signaling promotes the development of androgen-independent prostate cancer in human. And also GRPR, some studies found to be expressed in the vast majority of lymph node metastases and in about 50 to 70% of bone mets in prostate cancer. So what is AMT? So because my research will be focusing on the expression of AMT genes, so I'm just highlighting this process as an important process in prostate cancer metastasis. EMT is not standing for emergency medical technician. It's not the abbreviation that I wanted, but it's epithelial mesenchymal transition, which means that the epithelial cells acquire mesenchymal phenotype. So what are the characters of epithelial cells? What are the characters of mesenchymal cells? So for epithelial cells, there is cell-cell contact, which can prevent the cell from migration and invasion. While in mesenchymal cells, there is no cell-cell contact. The cell assumes the fibroblast in morphology, and it can invade and migrate. We have some markers for mesenchymal phenotype and other markers for epithelial phenotype. For mesenchymal markers, vimentin, snail slug. For epithelial markers, example E-cadherin and P-cadherin. So we came up with hypothesis based on this information that GRPR signaling is very important in prostate cancer proliferation, invasion, and metastasis of prostate cancer. We had some objectives. So we did in vitro study. We are currently doing in vivo study. For our in vitro studies, the objectives were to determine the impact of this signaling on tumor cell proliferation and migration, also to evaluate the expression of AMT genes in case after induction and blocking of GRPR signaling. For our in vivo study, we'll determine the effect of using an agonist like bombesin on the growth, invasion, and progression of prostate cancer in old mice. So for our material and methods, we use two cell lines. The first line is ACE1. So ACE1 is a novel cancer cell line that has been developed in our lab. And it's one out of 3 cell lines that have been developed in our lab. And we found that ACE1 has the lowest canine GRPR expression. Remember that, has the lowest canine GRPR expression. And in collaboration with Dr. Tweedle's lab, his lab actually was able to transfect ACE1 with human GRPR, and they create multiple clones of this cell line, and in our lab we tested these clones for ligand binding and for human GRPR expression, and we found that clone 15 has the highest human GRPR expression. So ACE1 GRPR has been transfected with human GRPR. So for methods, we did cell proliferation assay, wound healing assay. We did real-time PCR and statistical analysis. We used 2 agents, one of them to induce, which is bombesin. It's a 14 amino acids homologous to C-terminus of GRP, which is a natural ligand. And so C-terminus is highly conservative region of GRP. And bombesin binds with higher affinity to GRPR. We used antagonist to block GRPR signaling, and it's a compound that has been developed in Dr. Tweedle's lab. So we did cell proliferation assay, as I mentioned before, and we used 3 different combinations of the cells. So we used just ACE1, which has very low amount of canine GRPR, and ACE1 GRPR without any addition, without bombesin or without agonist or antagonist, and ACE1 GRPR plus bombesin as agonist. So theoretically, you should see that GRPR signaling is positive in the 3rd group. We use solometer to determine the cell number and viability, and most of our cells were viable at this time. So for growth curve, we seeded the cell. In each well, we seeded 1,000 of the cells, and we harvested the cell at day 3 and day 5. So if you can see, there is no difference at day 3, but at day 5, you can see there is a statistical increase in the proliferation of ACE1 GRPR plus if the bombesin is added. So we can see that in the group, if ACE1 GRPR plus agonist, there is higher proliferation rate at day 5. Also, we did wound healing assay. So in order to do wound healing assay, you have to bring your cells to 100% confluence and then make a stretch using 200 microliter pipette tip along the middle line. This will create 1 millimeter wound in your cells. So our results showed that after addition of bombesin as an agonist to ACE1 GRPR, you can see that there is enhancement in the wound closure. So after 24 hours, if you compare both ACE1 without anything and ACE1 GRPR plus agonist, there is enhancement here in wound closure. So here it's about 1% of the wound, and here it's about 13%. We started with 37% of the wound. By the time you can see after 24 hours, here it's 13%, and here it's 1%. So this means that bombesin enhances the migration and wound closure in ACE1 GRPR. So this graph representing the previous figure. So you can see we started at day 0. The wound is about 37%. Along with the time, you can see there is a statistic decrease in the wound area in both ACE1 GRPR, it may be due to canine GRPR, and ACE1 GRPR plus bombesin. But after 24 hours, you can see the dramatic decrease in the wound area in case of ACE1 GRPR plus bombesin. Also, we did real-time PCR. We did bombesin time course experiment. So we used 3 6-well blades for ACE1 and 3 6-well blades for ACE1 GRPR. The first well seeded with negative control for each cell. So the first well is ACE1 only here, and here is ACE1 GRPR only. And then we add bombesin for different times for 30 minutes, 1 hour, 3 hours, 6 hours, and 24 hours for both of them. Extracted RNA and then doing real-time PCR. So just the difference between negative control, just ACE1 and ACE1 GRPR without addition of agonist, we saw difference. So you can see that ACE1 GRPR has higher twist, VEGF-A, and TGF-beta if compared with ACE1. And we found that GRPR signaling upregulated some AMT, mesenchymal markers. So I'll just mention some of them. For the first one, Vimentin expression, we found higher VIM expression starting from 1 hour, lasted for 24 hours after addition of bombesin to ACE1 GRPR. And there is tendency toward higher VIM expression in ACE1, but it wasn't statistically significant, as you see. Okay, so for slug suppression, the same thing. We found that there is higher slug suppression starting from 6 hours and lasted for 24 hours. And in ACE1, we just saw a small trend for higher slug suppression after 24 hours, but it wasn't significant. For twist, also there is higher twist expression started from 1 hour and lasted for 24 hours after addition of agonist to ACE1 GRPR, while in ACE1 there is no difference between non-treated and treated cells. For snail expression, we found that there is higher snail expression starting from 3 hours and lasted for 24 hours after addition of an agonist, which is bombesin to ACE1 GRPR. But in ACE1, we found higher snail expression started from 3 hours, but after that, it decreased, as you see. We use antagonists to block GRPR signaling. And we found that after blocking of this signaling pathway, this induced MET phenotype, which is the reverse of AMT. So the mesenchymal cells turn to epithelial cells. So for them, as a mesenchymal marker, we found lower VIM expression starting from 3 hours and lasted for 24 hours after addition of antagonist to ACE1 GRPR. The same thing here in snail. We got lower snail expression starting from 6 hours to be dramatic decrease after 24 hours after use of antagonist. While in E-cadherin, we found the opposites because it's epithelial. There is increase in E-cadherin expression starting from 1 hour of addition of of antagonist, and lasted for 24 hours. So we are doing in vivo study in these days. We tried intratibial injection. We didn't get the result yet, but we will use different injection, sub-Q injection and intracardiac injection. We will divide the nude mice into 3 groups. One of them will be injected with ACE1 Luke, and 2 of them will be injected with ACE1 GRPR Luke. So one of 2 groups will be then injected with bombesin as an agonist. So theoretically, the 3rd group will be positive for GRPR signaling. And Luke means luciferase. So these cell lines has been transduced with luciferase, and by injection of D-luciferin, this reaction will emit light, and then the light will be analyzed by using IVIS machine. So we'll do IVIS imaging to see biodistribution of the tumor cells and also to record the photon per second. Also, we will do histological studies and radiographic imaging. For summary, so we found that GRPR signaling promoted cell growth and enhanced the migration and invasion of tumor cells, and also activation of GRPR signaling resulting in upregulation of some AMT mesenchymal markers and downregulation of epithelial markers. Blocking of GRPR signaling induced MET, mesenchymal to epithelial transition phenotype, through downregulation of VIM and snail and upregulation of E-cadherin. Conclusion, the GRPR signaling is very important in proliferation and the migration of the cell, and is likely important in prostate cancer metastasis. And targeting of this signaling pathway might be a promising strategy for treatment of prostate cancer. Our future plans identifies downstream molecules of GRPR signaling in prostate cancer, and also to determine if the GRPR signaling has any role in the vicious cycle of prostate cancer in the bone. Also investigation the relationship between GRPR expression and androgen receptor activation in prostate cancer. By this, I'd like to finish by acknowledgement of my PhD committee, Dr. Russell, Dr. Dwell, Dr. Tribune, and Dr. Shabsi. Also, I'd like to thank everyone in our lab, especially Wessel. He taught me a lot of things. He taught me all molecular techniques. Also, thanks for Jess for teaching me how to do IVIS imaging and Faxtron analysis. Thanks for Ethan, Washi, Bardis, James, and Lucas for being helpful for my experiment. And also, I'd like to thank Dr. Tweedle Lab for supplying us with ACE1 GRPR. By this, I'd like to say thank you, and I'm very happy to receive any questions. So bombesin is activating GRPR receptors, and then this will lead to activation of the signaling pathway underneath.
Okay, so do you know that that's specific? Because, I mean, there's a lot of papers back from the 70s and 80s that bombesin had oncogenic activity. Induces proliferation, it turns on a lot of pathways. So have you tried any other activating proto-oncogenes
Yeah. So the question is, did I try other agonists than bombesin in induction of GRPR signaling, right? Okay.
Or not. You know, I mean, there are other cell proliferative, there are other compounds that will induce the cell to proliferate, and similar to and proto-oncogene activity, and you may get the same kind of effect on these wound healing type assays that may not be directly related to the receptor.
Oh, okay.
Yeah.
So I didn't try any agonist other than bombesin, but according to literature, they use bombesin to induce GRPR signaling because it has a C-terminus, it's homologous to the C-terminus of GRP, and I didn't see any study using other than bombesin in this induction. The other question, it may be the bombesin activate other signaling pathway other than GRPR, right? So we used negative control in our study to see that. So if it activates this signaling, but other signaling pathway other than GRPR signaling, it will lead to induction of the expression of AMT genes to be higher in ACE1, for example, or ACE1 GRPR. So we made two negative control. It should be one ACE1 has a very low amount of canine GRPR, and ACE1 GRPR with human, it's ACE1 with human GRPR. So if that is the case, we should see higher expression of AMT genes in those cell lines.
And does the
GRPR, it's very hard.
Would it be on the cell surface?
Yeah, so
Cell surface?
it's the GRPR is on the cell surface. So GRPR, it's in the cell membrane. Once the agonists bind to GRPR, undergo internalization and be recycled again to the cell membrane.
And so can you block that? Can you block the whole process with antibodies?
Yeah, it may be a good idea.
Or you haven't tried?
No, I didn't try. Oh, ACE1. So yeah, is ACE1 androgen sensitive? Your question. Okay. So ACE1, we tested androgen receptor. We tested the expression of androgen receptor. We see that ACE1 has a very low amount of androgen receptors in terms of expression. Yeah.
Questions?
Any question from Worcester? Okay. Thank you.
Thank you very much.
I'm good. How are you doing?
You're still on, I don't know if you're on.
Do you know if, so I had something interesting happen when we tried to log in for the faculty seminar
Okay.
on Monday.
Mhm.
So, and I haven't gotten a response back from Fletcher.
Okay.
That's not a good sign.
But, and it may have been something that I did because I logged in and I did something on my iPad and then I logged out. And do you know if you have to be added as a
Yes.
host